DNA fingerprinting is the approach of determining an individual's DNA characteristic. It is invented by Professor Sir Alex Jeffreys in 1984. DNA fingerprinting has been used to simplify criminal identification by using DNA found at crime scenes to identify possibility of the individual's involvement in the crime. It has also been used in parentage testing, established immigration eligibility, studies on animal and plant populations, and in genealogical and medical research. The gene coding for a particular protein contains 95% non-coding DNA and only 2 to 5% coding DNA. The non-coding DNA can be categorized as single copy spacer DNA or multi-copies DNA, also known as repetitive DNA. The repetitive DNA sequence can either be mini-satellite DNA, which is variable number tandem repeats VNTR for longer blocks of sequence, or microsatellite DNA, the STR, short tandem repeats. There are two types of DNA. First is the genomic DNA, which exists in the nucleus as chromatin. Teeth is a good source of genomic DNA. Second is mitochondrial DNA, which is the small circular chromosomes found in mitochondria. This mitochondrial DNA can only be passed from mother to offspring. It is used when the DNA sample obtained is insufficient or degraded. Stages in DNA extraction begins with cell membrane rupture, next proteins denaturation by chelating agents, and inactivation by proteinases. And then lastly is the DNA extraction by several methods such as organic method, Chalex 100, FTA paper, or isopropyl alcohol. The organic method DNA extraction is commonly used in forensic dentistry. This technique uses phenylchloroform, which is used for extracting DNA with high molecular weight. However, this method is laborious, time-consuming, has higher risk of errors, and can only be done if there is abundance of sample available. Chalex 100 is a chelating resin. This is the fastest method for DNA extraction. It has the lowest risk of contamination, but it is expensive. DNA extraction using FTA paper is composed of adsorbent cellulose paper with chemical substances. This speeds up the use, making for quick extraction. DNA extraction using isopropyl alcohol contains ammonium and isopropanol. This method is less expensive, and it is also an alternative to the organic method.
Next, there are various methods available to run extracted DNA for further analysis. First is PCR analysis. The principle of PCR analysis is that a single copy of DNA is amplified into many copies with the aid of enzymes and primers. The process of PCR includes denaturation, which is unbinding of DNA, and annealing, which is the annealing of primer to the target sites, and extension. After 2 hours, DNA amplified 109 times to the original amount through 30 cycles, and amplicons are detached through gel electrophoresis. The application of PCR includes the evaluation of variable number tandem repeats of DNA samples and to quantify DNA of different genders in mixed sample, especially in sexual assault cases. The next method is restriction fragment length polymorphism RFLP. The principle of RFLP is that the restriction enzymes identify and cut at specific sites of genomic DNA. The first step in the process of RFLP is that the isolated DNA cut into DNA fragments using restriction endonuclease enzyme. Fragmented DNA has variable number tandem repeats P and TR. Then gel electrophoresis separates previously cut fragments based on size. Then Southern blotting transfers fragments to nitrocellulose membrane. After that, probing allows radioactive probe to bind to DNA. Then, X-ray image of DNA obtained through autoradiograph. Finally, the identification of DNA fingerprint of a person by detecting unique VNTR pattern will be done. The cons of this method includes: firstly, it requires large samples. Then, it also requires long waiting time. The third method is short tandem repeat STR typing. The principle is that it has high power to differentiate individual due to its highly polymorphic, non-overlapping size of alleles for about 2 to 7 base pairs in each repeat. The process of STR typing includes gel electrophoresis, silver-stained polyacrylamide gels in those days, capillary electrophoresis, and ABI-based gene sequencer, where all this employs fluorescent detection method. The application includes: it is used as a marker in forensic investigation, where it serves as a standard for CODIS system with 13 definite STR loci, which is known as CODIS markers, and sex identifying amylogenin marker. Besides, it is used in paternity testing, as each person has some STR inherited from both parents. STR is also used for mass victim identification during calamities from the DNA sample extracted from tissues such as bones or teeth. The next method is mitochondrial DNA mtDNA analysis. The principle is that every kid has identical mtDNA as mother, since embryo's mitochondrion is solely from mother's egg. The process includes direct sequencing of nitrogenous bases, and the cons of these methods are limited information, since it's only involves maternal familial relationship, and also it is very expensive. The application includes it is used if nuclear genome analysis is not feasible from ancient tissues like hair, teeth, and bones. It is also used to identify missing person by comparing mtDNA of anonymous remains to a possible maternal relative. The next method is Y chromosome analysis. The principle is that every male child has Y chromosome passed by father. The Y chromosome region has specific STR which is useful for investigation. The process includes targeting YSTR using primers, and the application is it is used to find out relationship among males. The next method is single nucleotide polymorphism SNP analysis. The principle is that the alteration in nucleotide sequence provide useful information for analysis. The application includes it is used to provide vital information on descendant, gender, and evolution. The pros includes it is highly automated and could identify highly degraded DNA fragments.
DNA fingerprinting is one of the most trustworthy techniques that being used in solving the crimes and others. One of the most applied fields of DNA fingerprinting is in the criminal investigation. Other than that, partner disputes can also be solved using this technique. Furthermore, DNA fingerprinting can also help in dropping the crime's blame on innocent people. There are several sources of DNA in our body, so tooth is the best source of DNA extraction. It is because teeth has a hard tissue and closed palm that helps in protecting the DNA from harsh environmental conditions that can harm the DNA. Thus, the DNA in the teeth are very valuable. Dentin and palm are the places where the DNA can be extracted in the teeth. The techniques of DNA extraction that can be used are sectioning of teeth horizontally at the cameto-enamel junction or vertically up the root tip, scraping, and aspiration. On the other hand, DNA extraction also can be done by crushing the teeth, cryogenic grinding, or conventional access cavity preparation. Access cavity preparation technique has many advantages, such as simplicity, has a lower cost, and the tooth integrity can be protected. Furthermore, DNA extraction process can be divided into 3 stages, which are cell rupture, protein denaturation, and activation by using chelating agents and proteinases, and DNA extraction. The most used technique in forensic dentistry is organic method DNA extraction that use phenol or bicylic 100, FDA paper, or isopropyl alcohol. Other than teeth, human saliva also can serve as a very good source of high molecular weight of DNA. Saliva usually obtained from material scenes such as clothing, cigarette butts, and envelope stamps. Besides that, it also can be obtained from a bite mark. In order to get the DNA, the skins was swabbed to collect the saliva, and this swab will be tested for amylase. So, a positive test results will show that there is presence of saliva and the injury is a bite mark. In conclusion, teeth is a unique element and it is one of the best sources of DNA extraction because it is resistant to destruction. So, the dentist should not only have the knowledge but also must have an awareness on the importance of forensic dentistry and its applications.